step or another by oxygen. That's what's really important. So if you look at other carbon dioxide fixation pathways, like the ones for methanogenesis, many of the enzyme steps are very oxygen sensitive. They won't work. And a lot of the other anoxygenic photosynthetic bacteria actually have very oxygen sensitive enzymes. So I think the evolution of the Calvin-Benson cycle was really critical in this before you can even start oxygenic photosynthesis. And there's also a kind of an earlier form, perhaps, of cyanobacteria called Prochlorococcus, which actually is a little bit more primitive and doesn't have the range of photoactivity. And I'll make another point about nitrogen fixation. All the evidence points that the origin of nitrogen fixation was in methanogens. Methanogens invented nitrogen fixation, and then it got transmitted through a variety of bacteria, but never to a eukaryote. And probably the reason why it's so oxygen sensitive is because it was invented in an oxygen less environment. And a lot of you have asked me questions about whether or not water could be formed biologically. And what is really interesting in this same topic is that the way microbes one of the ways microbes handle getting rid of oxygen at sites where you get oxygen, like nitrogen fixation, is to produce an enzyme called hydrogenase, which takes oxygen and hydrogen and makes water. And how extensive was that in the first 2.5 million years, where you probably had oxygen from photolysis and oxygen radicals? How much water would have actually gotten produced under those circumstances? Was it significant? I don't know. I never thought of that. It was some of you and students here that actually brought that topic up. So it's an interesting one to look back in evolution and see when various processes emerged and why, and how they were essentially adapted to environmental conditions. So where am I? In keeping with my tradition, I'm showing this because it's newly discovered, and it's only been in the last few years that we've actually found carnivorous feeding of sponges. Normally sponges are filter feeders, so they take in suspended material and filter them down. In hydrothermal vents, a group of the French found in early 2004, 2005, a group of carnivorous sponges. These are absolutely gorgeous sponges, and you can see them. I don't know what these little balls are filled with, but frequently they're filled with carbon monoxide on a lot of other species. These are what we call demisponges, which are siliceous. And you notice they're on basaltic rock. These are hydrothermally environments. And this is another group that was very recently found along some back arc basins, which we're just starting to go to. And these are like little puff balls, but they're sponges, and you can see that they actually have these tentacles around them. And these basically are like Velcro, so that when an animal swims into them, they completely are stuck. And once they're stuck, then they're consumed by the sponge. So these are the first, and this will give you some idea of the amazing kind of strategies. These are small. These are depth. These were discovered in the 2006 cruise, one of the 2006 cruise. So these lip-shaped spicules basically they're like sticky Velcro, and they catch prey. But it's really interesting. Sponges don't have a gut or a mouth or anything. They're individual cells, the same kind of cell that comes together and forms a structure. So they're some of the most primitive in that respect for multicellular kinds of eukaryotes. And what's interesting, without a mouth or stomach, the cells in the sponge kind of all the cells stream towards the prey, so that most of the cells then in the sponge get a little bit of the nutrients from that. A really interesting strategy. well, I like showing these things. So anyway, there's something I hadn't planned on doing until yesterday, last night, is I'm going to highlight, after I finish a couple of slides on low nutrient environments, I'm going to highlight a paper that came out just in 2010, which has extreme significance to it as far as I'm concerned. It's the first multicellular animal that grows in the absence of oxygen and is totally adapted to that. We have single cell eukaryotes, protists, or like protozoan, that can grow anaerobically, but we've never found a multicellular organism. So this was the very first. And I thought, in thinking about where evolution can go in the absence of oxygen, this might be actually a fun kind of animal to explore in that respect. And then I'm going to start Origin of Life 1, and that'll be a sort of a continuum of maybe 3 lectures or so, because I'm going to alter this to, I think, respond in a biological way to some of the really wonderful things that Jim has talked about and Jonathan has talked about, and talk about how I think microbial metabolism fits in within the changes in atmospheric chemistry. And also what I'm going to try to do is predict what these new metabolisms, how they might actually influence the upper atmosphere, because I had a very nice discussion last night over dinner on what we might actually be able to look for on exosolar planets. So this is a highly evolving adaptation in what I plan to talk about early on. So I want to introduce you to what are called oligotrophic organisms or oligotrophic environments. And Jim mentioned cyanobacteria in the ocean, where we see cyanobacteria dominate as the photosynthetic organism, dominate over larger eukaryotic phytoplankton like diatoms. These are usually environments that have very, very low nutrients of one kind or another. And so some of the most common microbes in the open ocean and in alpine lakes, for example, grow on microgram levels of organic material, extremely low, and they're very well adapted to do this. This group of microbes is really the reason why we're interested in Lake Vostok, for example. We want to see if there's some interesting oligotrophic microbes that might actually tell us something about what could live on an icy moon with a liquid ocean. So that motivates this kind of research. There are other microbes that grow just on carbon monoxide and methane. And just last year, published in Nature, is the first microorganism that can actually grow anaerobically with hydrocarbons. This is very hard to do because hydrocarbons are very reduced forms of carbon. And how do you actually oxidize them in the absence of oxygen? And it's an interesting paper that I won't go into. I'm just going to highlight a couple of different kinds of cells. Colobacteria, which are called prothecate because they have this attachment. So they attach to surfaces with this little appendage, and then they make daughter cells with flagella that swim around and then attach somewhere else. I'm showing this because, I used to keep jars of distilled water in my lab for, biochemical analysis, and wondering why nothing really worked very well for me. And if I scraped the inside of the glass vessel of distilled water, I would find these guys in it. They can grow on extremely low levels of nutrients, very slowly, but they do grow. And they're commonly found in really low nutrients. SAR-11, SAR named after Sargasso Sea, which is the first time this group of microbes were identified in 1990. Where the first isolation of SAR-11 came about just a few years ago, and it's called Pelagibacteria ubique. It's the most numerous bacterial species in the world, estimated to be about 10 to the 28 cells. So there is it is clearly it rules the world in terms of the most abundant microbes. Now apparently it has more than one kind of physiology, but it grows in micromolar levels of organic compounds. And if you take dissolved organic material in the ocean, which is in very, very low amounts, it will grow on that. If you double that, if you concentrate the dissolved organic material and give it to them, they don't grow. It's too high. So they are very adapted. And what they have are binding kind of proteins on their cell wall that have an affinity to very, very low levels of a variety of organisms. It can eat DMSO, dimethyl sulfide, and it also, just recently, in a genome was found to have this pigment like we have in our eye that might actually derive some energy from light. We don't know that yet. So that's all I want to say about that. But that's the kind of work that most of us in biological oceanography have to work on, are microbes that grow in really low nutrients. So this is the first anaerobic metazoan. And so first, why do metazoans like oxygen? And that's because mitochondria. And Jim mentioned chloroplasts, which are in eukaryotic algae, and as a result of endosymbiosis, well, all eukaryotes have generally mitochondria, and that carries out respiration and oxygen production and energy production. And that carries out in a mitochondria. The mitochondria are also bacteria, and they still have DNA, just like the chloroplasts do. And we can root precisely where the mitochondria are. They're called alpha proteobacteria, and are not really super ancient, but probably somewhere around 2.5 billion years ago. And we know a lot about the mitochondria. And just to give you a little hint of this, its structure, it has an inner membrane, and then an outer membrane, and then the crista, which are these compartments, which give it a lot of surface area. The surface area is where all the enzyme activity is. And then the matrix around the crista contains hundreds of enzymes, ribosomes, the mitochondrial DNA or genome, and the tricarboxylic acid cycle, which is the dominant cycle for producing ATP. So for example, the TCA cycle, this cycle is so efficient, if you take one glucose molecule and run it all the way through, you theoretically can make 38 ATPs out of that. Generally, you don't make that many, but you can make that. So it's a very efficient energy type process. Usually a cell has hundreds or thousands even of mitochondria, and they can represent up to 25% of the cell's cytoplasm. That's us. We're very dependent on mitochondria. If you actually we all meditate and close our eyes, we can actually feel their activity in our cells. That's the problem with being a microbiologist. I'm too aware of all the microbes in me, and I have to just not think about it. And you over here with a beard, I just look at that beard and I just see this massive garden of microbes. And I can give you a whole lecture on beard microbiology, but I'm not going to do it. Well, it has astrobiology implications because, hey, when they were going to send men up to the moon, NASA was really concerned, should these humans shave when they go up? Is this okay? Can they grow a beard? So they actually did some studies, and as the beard grew and all these microbes were falling down all the time, they said, you better shave. But out of that came a couple of publications, and since then there's been some other publications on, it's actually, it's kind of cool. You are really, it's like having a little garden on your own face. And so, and we males can do that, so it's kind of cool. So, but it's actually more fundamental about mitochondria. Bill Martin at the University of Dusseldorf is probably one of the best researchers in this field of mitochondria and the origin of mitochondria of anybody, and has some really interesting theories about it. But he points out in this 2002 paper that biochemistry textbooks depict mitochondria as oxygen-dependent organelles. But we found many mitochondria can produce ATP without oxygen. And in a paper that came out in 2010, also by Bill Martin, he actually has an origin of the very different kinds of mitochondria. And and what they actually do. So while they produce ATP, they're not necessarily all have to be oxygenic. So this takes us back to the Donavero paper that was published online on an online journal just a number of months ago. And part of the reason why this paper was published in this journal and not in Science or Nature, because there was a lot of competition to get this out fast, because there's a lot of people very interested in this topic. What they did is, I'll show you, they went into, this is the Mediterranean, went into some really deep sea areas where there are these brine pools, very, very high amounts of salt in these brine pools, very deep, more than 4,000 meters deep in the Mediterranean. And I'll show you the information in that. And it's not only very, very high salt, but it has no oxygen at all. And in looking through the sediments, they found an animal called a Loricifera animal, which is new to science. In fact, this whole group of animals is fairly new to science. And they used a variety of methods, which we'll go into, to show that it was actually active and growing under these conditions of no oxygen. But they also found it lacked mitochondria, but had a different kind of organelle in it, and I'll mention that. So this is the site in the Mediterranean. Its depth is almost 4,000 meters. The brine layer depth, where the little pool is about 400 meters. And the salinity, we in oceanography talk about parts per thousand, so this is 36% salt. This is very, very high salt. Temperature, high hydrogen sulfide, relatively high methane, and high ammonia, so all the sorts of reduced power in that. In the Gulf of Mexico, this is one of the best studied brine pockets. These are caused by diapirs, where you actually have deep subsurface pools of high saline that make their way up into the ocean floor. And there's a lot of very interesting science that's done around this, but we don't normally see a lot of animals hanging out on these high brines. We see them around the edge, mussel beds around, because there's also associated with those a lot of hydrogen sulfide. And so the mussels here have endosymbionts that oxidize the hydrogen sulfide, and they make a living that way. So this is a Loricifera, really cool animal. It's small. It's up to a millimeter or so. It dwells in sediments. There are 22 described species and 8 genera, and a lot by the French that haven't yet been described. Most of this is a result of the census of marine life going on the last 10 years. They attach themselves to sediments. They actually have a head and a mouth region and a whole digestive system. They don't seem to have a circulatory system. And they have a relative, for this size animal, a relatively large sized brain for whatever they want to use it. And they have a fairly, I don't know, what the brain does in these things. But this is the animal stained from this environment. This is the life cycle of these kind of animals from the small larval forms up until the adult male and female. So they're pretty elaborate metazoans. They're not minor items. Actually kind of cool. So the evidence that these things grow in the absence of oxygen, there's a series of stains that we can use that can indicate whether or not something is alive or dead. And they used those. They also did electron microscopy and other kinds of microscopy and determined that there was no mitochondria. And then they did some isotope uptake and other biochemical analysis. And again, looking more at these with live stains versus those that are dead that actually don't pick this sort of thing up. And one of the other pieces of evidence is that there's no evidence of attached microbes. Usually when animals are dead, microbes immediately attack them and degrade them, and they didn't find that in this. And this is a live dead stain. And if you look at this, it's a series of images across different sections of the body volume of this particular animal. And so what these represent are progressive scannings from the inner to the outer part of the body. And the cell tracker green, we call it, is the live dead stain. And then in B, these are the animals that were killed by freezing, for example, and they don't take up the dye. So this is the take-up of the live stain, which is an indication that they were metabolically active. And I won't go into this other than there's an increase in isotope uptake in these animals and in comparison to the dead animals. What's really interesting is they don't have mitochondria. They have these structures which they're calling hygienosomes. And within these hygienosomes there appears to be microbes that look like archaea, and that's what apparently they are. So there appears to be endosymbiotic archaea within these hygienosomes, and they appear to be methanogens. So it looks like what this particular organelle is something that takes up hydrogen and CO2, makes methane, and contributes something to the growth of these animals. And so what I want to do is use this. We're so generally fixated on mitochondria, but there are really several different organelles. So there are mitochondria, which can function both with oxygen and without oxygen. There's these hygienosomes, which I'll mention, which have previously only been found in single-cell animals that grow in the absence of oxygen. And then this newest one that we don't know very much about that are being called mitosomes, also found in animals that grow in the absence of oxygen. And so I just want to point out, this is from a very recent paper in PNAS. It's a model of ATP synthesis, and you note this pathway, from hydrogen to possibly CO2 or pyruvate. It's hard to say, but the intermediates are acetyl-CoA and acetate. The methanogenesis pathway, the main intermediate is acetyl-CoA. What this is we think to be the earliest high-energy organic compound and maybe the driver of early life. What this is a carbon-sulfur bond, and the carbon-sulfur bond has a lot of high-energy potential. And this is what drives methanogenesis. And it also drives acidogenesis, which has the same pathway. So rather than make methane, they produce acetate. And ATP is driven. So this really looks like some very ancient form of the kind of pathway that's involved in making methane that's inside of this particular animal in the Mediterranean. So I'll leave it with that and just do a summary of extreme life. So only high temperatures, max I don't think we know, and low temperatures probably less than -20, and the absence of sufficient water prevent growth of microorganisms. It's something we talked about. Microbes can utilize a wide range of inorganic and organic compounds and elements as energy sources. The last time I calculated it, there are literally thousands of different organic compounds that can be utilized as sole carbon sources by microbes. Microbes have adapted to grow in ultra-low levels of organic and inorganic nutrients, and then multicellular animals have been shown to grow at 50, and in the absence of oxygen. Those are the new findings. It is very likely that there are planetary bodies, and particularly icy moons with oceans, that can support Earth life. The question is acquiring such life. I would like to keep hitting on that. So this is where I was supposed to start today, which is an origin of life and early evolution. And this is going to take me through the next probably 3 lectures, I expect. And as I progress through this, I'm actually changing this a lot from what I had actually intended to talk about based on what has come out of this discussion and out of, I think, this really wonderful talks that Jim and Jonathan have really stimulated me in rethinking even what I had. I wish I hadn't heard them. My life would be simple here. But now I have to change the way I think. So anyway, the outline, I'm going to give you some history, which is the history of the origin of life is really fascinating and interesting. And then these questions of when, where, and how, and what are the questions that I will deal with. So really, when in Earth history, and Jim mentioned that, certainly older than 3.5, but actually there's some pretty strong evidence at 3.8 in the Yeshua. And where did life originate? That's a big question, because should we have everything go on in one kind of setting? So is all these processes leading to life and life itself go on in one setting? I don't think so. And so the where really means perhaps where did the different steps leading to the origin of life take place on an early Earth, and then what subsite of that actually was where the first microbial community. Then how did life start? This is something we really don't understand, but I'll talk a little bit about where people are going with it. And would an understanding of when and how questions help constrain the settings where? This is kind of a top-down approach. If we can come up with some theory about how and see something in the paleo record, can we infer exactly where early life started? And then, of course, what did the earliest life look like? And I think we can talk a little bit more positively about that. So a little bit of historical perspective. I'm going way back now to the 6th and 5th centuries before Christ. When this came out, I'm a history buff, so I can't help myself. Organic life could and does arise from inorganic matter. This is generally attributed to Anaximander, which is a Milesian philosopher, now Turkey. And so this idea of spontaneous generation, that from nothing came something, came very, very early. Aristotle, life arose from the four elements, earth, air, fire, and water. Since Aristotle denied that the universe and the earth had a beginning, life occurs all the time. So he just got out of that whole thing. You know, I wish he hadn't, because I would have loved to have heard his speculation on the origin of life, but that's it. So the idea of spontaneous generation persisted into the 19th century, and if you look at the history, is wonderful. There's literally hundreds of recipes for making life. I'll just show one of them. If you take wheat grains, put wet rags in them, in an open jar, mice come in 21 days. And so this is how spontaneous generation works. Now, there's a huge number of recipes for making things like maggots, you know, and so there's probably 100 different recipes from, I mean, I get maggots in the flour that I get to make bread. I open it up, I don't know, all these things are moving around. So anyway, so that's how this idea persisted of spontaneous generation. So I mentioned the end of spontaneous generation or not. I'll say something about this. Well, there's this rich history, particularly in the 17th and 19th century. Much of the idea is centered around abiogenesis of animals. So we're not thinking about microbes. We're thinking about how to make animals. And so in the 17th and 18th century, Francisco Redi and Spallanzani and John Needham performed experiments with open and closed containers showing that animals didn't appear in these closed containers. So, I mean, and these were big things, you know, these were big experiments, but still it did not persuade the community at all that spontaneous generation couldn't occur. So the definitive experiment was formed by the illustrious microbiology Louis Pasteur, for which we name pasteurization for. He was the one who did the first vaccine for rabies. He made lots and lots of different, in fact, he was one of the first to show chirality using tartaric acid in wine, for example. You know, that sometimes you open up a bottle of wine, you have these crystals in there, they're tartaric acid, and there's D and L forms, and he was the first to actually show that. So anyway, what Louis did is performed a really interesting, simple experiment. Took a flask, and he put the tip of the flask under a flame so he can bend it and then twist it out. And he took, this is called a swan flask now, and he put a culture medium for microorganisms in it. It's basically a beef broth. is what he had, that microbes like to grow in. And so he took 2 of these, and one, he put it in boiling water for a couple of hours, and another one he didn't. And then he just left them at room temperature. The one that he didn't put in boiling water turned turbid, filled with microorganisms. And the one that didn't. And the reason is just by gravity, contaminating microbes from the air couldn't get up through this. And so the microbes that were there prior to heat sterilization grew. And so this experiment really best disproved spontaneous generation, and we saw the beginning of the fading of that concept. But the reason I mentioned the end of spontaneous generation or not is, really did Pasteur prove that life could never come from non-living matter? What he did disprove was that modern, contemporary life, less highly evolved, living organisms arose from non-living stuff. So this is very different from the idea that the first primitive life once arose from a non-living, non-organic kind of material. It's very different. Obviously, we've had some kind of spontaneous generation to get life, just not something that happens in 21 days. So Charles Darwin is only in only the first edition of The Origin of Species, and I'll mention why, wrote, only in the first edition, I should infer from analogy that probably all of organic beings which have ever lived on this earth have descended from some common primordial form into which life was first breathed. He had to remove this because of the Victorian mores of the time in England. This was heretical for him to say this. So in the subsequent editions of The Origin of Life, of The Origin of Species, this quote was removed. But it also shows where his head was. His head was in thinking about, the origins. In fact, in a letter that he wrote to one of his colleagues, I also attribute The Warm Little Pond to Charles Darwin. He actually sent in a letter this tree of life. And look at it, it actually has a root to it. He talked about this and branching. So this is the first tree of life that I know of, and it was through a letter. So he was already thinking about how descent would happen, but all from some common ancestor. And he wrote this to one of his colleagues. In The Origin of Species, he actually does include these great trees of life, and note the branching and the roots, and so he was way ahead of his time in determining how actually evolution was really going on, and really thinking very much about deep down in here, but only through his letters and not publicly. So the modern era of The Origin of Life really starts in the earliest 20th century with Haldane and Oparin, and I'll talk a little bit about those guys. Haldane was a geneticist, Oparin was an organic chemist. Haldane in England, Oparin in Russia. The idea that life arose from organic material formed abiotically from inorganic material, including a replicator and metabolism, all came from Haldane and Oparin. And keep in mind, this is before DNA was found. And then, as Jim mentioned, Stanley Miller in 1957 demonstrated that amino acids could be synthesized from inorganic gases that he thought at the time to dominate the early atmosphere, which turns out to be wrong. Someone who rarely anybody thinks about today is Sidney Fox, because the Miller group and all that whole group did not like this person. And so this person had a really hard life, from what I understand. I mean, I was too young to know all this, but I heard about it later on. Stanley Miller really believed in sort of the warm little pond idea, and that eventually something that replicates came first. Sidney Fox believed that proteins were everything, that life started as some kind of a protein sphere. And the reason I mention him, he did some incredibly interesting chemistry. For example, he took amino acids and heated them under very specific conditions, and he made these spheres that look like cells. But what's most remarkable about this, when he took the same amino acids and did the same under the same conditions, he produced exactly the same peptide sequences. That is, the amino acids were all in the same place each time. It was a completely repeatable experiment. And I find that really profound in terms of trying to understand origin of life, yet it's sort of missed, I think, in the literature. And then, of course, in 1982, Thomas Keck reported on this discovery of catalytic RNA molecules, molecules of RNA that can actually behave like enzymes. And then he and others won the Nobel Prize for that later on. But out of that came the concept of the RNA world, which Gilbert published a paper in 1986 actually framing this concept of the RNA world. So this is the sort of background. I'll show you a picture of J.B.S. Haldane. He was trained as a geneticist, sophisticated man. He wrote a paper in the 1930s where he described the origin of life, and I tell you the truth, when I read it, I had goosebumps. That's how profound that paper is. And what was really interesting about it is that the year before he wrote the paper, viruses were discovered. So back in the early 30s, viruses were discovered, bacterial viruses, by independently by two researchers, D. Harrell and Twort. And so he was very fascinated in what the role viruses might actually play in the origin of life. And so when I read this, I don't know if I have it on my computer, but if anybody wants it, it's unbelievable. I mean, here's a paper that's 80 years old that is just so profoundly predictive of what we're doing today. An amazing paper. Oparin was trained as an organic chemist. And Haldane proposed that carbon dioxide, ammonia, and water vapor made up the bulk of the ancient atmosphere. So this is where Stanley Miller got his ideas. They're more from Haldane. And energetic radiation would convert these gases to organic compounds, their accumulation into an organic soup, which sprung life. This all comes from Haldane. Independently, Oparin proposed a similar model with the organic compounds forming coacervates, or essentially these little protocells made out of organic material, colloidal organic droplets. Oparin and Haldane believed that information macromolecule was a protein. This was prior to Watson-Crick and the discovery of DNA. So Oparin actually favored abiotic synthesis, in which an information-containing protein was the first step in life. By contrast, Haldane proposed that some form of metabolism was the first step. What's interesting is that today, this is the main debate. It's still replicator first, metabolism first. Now, if any of you know these guys, do you know what they had in common? Besides interest in the origin of life? They were both communists. And because they were both communists, they were totally interested in trying to find an abiotic way to make life and to dispel the concept of creationism. And so they were both driven by that same philosophy. They're both very interesting to read about, very interesting people. But this is Stanley, and Jim showed another version of this. This is Stanley, who passed away in 2007. And again, as Jim mentioned, his initial experiments were methane, ammonia, water, and hydrogen. And it was the first to produce a variety of amino acids using spark discharge, which was supposed to mimic lightning as an energy source. And I'll get back to what he produced later. So before I start, our paradigm shifting. The paradigm that life originated in an organic soup dates to Darwin and promulgated by Haldane, Oparin, and Miller and others, and is still a popular idea. So the idea that somehow you can get a soup and put 1 million organic compounds in it, and then bless it, Paternoster Filius Spiritus Sanctus, and out of it springs something really interesting. And that's where we are with that. So the soup people also favor, I say the soup people, you know, those of us who are pasta people, I guess, I don't know what we favor, but the soup people also favor the replicator first hypothesis. It's something like, they're the big proponents of the RNA world, for example. So during the past 20 years or so, a significant origin of life community has embraced the idea of settings, of looking at different environments, and hydrothermal vents are one of the ones that is being examined considerably. But those people favor a metabolism first model. And I'll try to, over the next couple of lectures, try to expand on that. So I know I'm running out of time, so I'll just run a couple of minutes then. So attempts to understand the origin of life, this is a complicated slide, but getting started, we need the prerequisite conditions for the origin and maintenance of carbon-based life: water, organic monomers, availability of key elements, catalytic minerals, environmental conditions. All of this start looking at the setting. Organic soup versus a focused production of precursors and catalytic organic compounds. That's the big controversy. Whether or not you start life with a million different kinds of organic compounds, or you find some catalytic mechanism to produce a lot of just key compounds. That is where the difference is between replicator first and metabolism first. So the metabolism first, the replicator first, and there's some peptide people. There's actually a sugar world group of about 3 people that really think sugars started, you know. So it's the sweet origin, and that's okay, you know. So they don't have a big following, but they've done some really interesting work on how to make sugars abiotically. My view is there's just one world, and so what I'm trying to do is how to bring all of this together, because there's no way that you can really separate metabolism and replicator. I think that you can't get a replicator without some form of metabolism that produces some kind of bond energy to drive the system. The top-down approach also shows a little bit of glimmer of insight. Essentially, the RNA world came out of the top-down approach, and out of that are an understanding of the origin of the genetic code. We don't know the origin of the ribosome. This is a really interesting a factory for making proteins. And we had a whole origin of life meeting that I went to at the Santa Fe Institute a few years back just on the origin of the ribosome. Extremely fascinating, but we just don't understand it, you know. So it's just it does have a bunch of RNA on it, but it also has, what, 20-something proteins associated with it. And those proteins are very, very key in translation and transcription. And maybe if I get a chance, I'll talk about the ribosome. And then this transition from the RNA world to the DNA world, and then taking all of this and matching it to some settings. Where did it start? Does the where have dimensions in space and time? How do we think about this? It's very, very complicated. And I know I'm running out of time, so I'm just going to end with this slide, which is a summary then of the bottom-up and top-down approaches. And what I'm going to do is go into detail on this in the next couple of lectures. So the bottom-up approach, again, is how do you make the organic precursors, and how do you either get them into metabolic circuits or into the RNA world, and eventually into a DNA life. And you start out with this. The top-down approach, again, we go to our tree. And I've inserted a tree from Shen and Buick that has things like dating for when they think photosynthesis occurred, and when the first eukaryotes at about 2.7 or so occurred. They placed this in this tree. But what we try to infer from extant life are what the settings for organic life would be, what the most ancient physiologies might be, ancient metabolisms. There's controversy whether it's CO2 fixing or organic utilizing. A lot of the soup people, obviously, if you're living in a soup, why do you want to fix CO2? So if you actually emerged in bouillon, in a beef broth with vegetables in it, why fix CO2? And so there is a controversy there. And then the RNA world actually came from a top-down. There are bacteria today that actually make these ribozymes, and that's where they were found. There's one called ribonuclease P, which is what won the Nobel Prize. It's a really interesting RNA. And then, again, early replicators. If I get a chance, I would really like to place viruses within a lecture, because I do think they're going to make a difference in our understanding the origins. So I'm going to end there, and then we're going to talk about, some of the specific topics associated with this. And then, so I can actually take questions now or even at the coffee break. I know Jonathan.
So I have a quick question about the top-down approach, and you're probably going to explain this later today. But there's a very, very large structure, a new sports palace that's being built near my laboratory in Rome. And it has this enormous space frame. And in order to build the space frame, which is self-supporting, they had to erect these two very large cranes, which provided tension for all the different pieces as they assembled it, so it wouldn't collapse until put it all together. Now those cranes will go away eventually, of course. So if you tried to take the top-down approach of understanding how that was built, how would you be able to infer that those cranes existed? That's my question.
It's a really interesting question, and I think you posed it in a very interesting way. Others have posed it in a different way. So in the top-down approach, what we ask is, what is missing? That's one way of handling it. But what we try to do in the top-down approach is really identify what is missing. And rather than try to identify it from first principles from a bottom-up approach. Do we know? I mean, we actually have organic compounds and life. That's what we have. Those are the two end members. And I think there's a lot of cranes in between those. It's not just one set. That's all that's all we can do. Are we missing something? Potentially, yes. Because what we're looking at is 4 billion years of evolution in a top-down approach. And so we make the assumption that those organisms that near the base of the tree retain genetic information that goes back to the beginning of life. That is an assumption that we make. And because of that, we tried to come up with the origin of metabolism, the origin of blah, blah, blah. And we try to then couple that with what we actually see in the paleo record. I mean, I can tell you when methanogenesis went on, so I think, Jim is right that it's ancient, but that doesn't tell you about the origin of methanogens or the organism themselves. I'm going to actually try to talk about that later on. And it'll be, but, I might as well show my biases. I'll let you know they're my biases so that you don't write them down and say, ha-ha, I now know how life started. But at least I have a model. Essentially, I've constructed some cranes of my own, and I'll show you what they look like. And you have.
Oh, I was going to say that there's another variant of the, I guess what you would call the soup hypothesis, where the organic compounds, instead of being produced in situ by lightning, for instance, they come in from interstellar space, and in meteorites and interplanetary dust particles. Yeah. And so that one's a little different because the organics that you get from there can be very complex. So it's a different twist on the whole idea.
Yeah, what we try to, you've taken me to the next lecture a little bit, but what we're trying to do is take a look at the processes on Earth that actually make organic compounds versus those that are coming in either from dust particles, comets, meteorites, shock synthesis, lightning, etc. What we do with those, what would we do with those if we got them? How can we make use of those? And I'm not sure that they're that important in the origin of life, but I will consider them. I'm more concerned with what Jonathan talked about yesterday, which is actually getting carbon in the Earth in any form that there is. So I want to make sure that we have carbon, but not necessarily the organic compounds. They don't really mean much to me, even though a lot of origin of life people think, haha, this is what we want, and a lot of this stuff is going to get into this warm little pond. And, you know, it's got everything in this soup. It just doesn't work. I had on the Santa Fe Institute meeting, I had a really interesting talk with a physicist who really very hard tried to model how you can get anything interesting out of a soup. It doesn't happen. He cannot model it. And the only way he can model anything happening is to have some source set of reactions that produces a lot of the key compounds. And then he can make it work. Then you have probabilities of making something actually happen. But your probability is just unbelievable in a soup to get anything to happen in it. So anyway, we'll continue on this discussion tonight.